The Joshua Boger Innovative School of the Year Award recognizes a Bioteach high school that distinguishes itself through outstanding educational programming in the life sciences. This award includes a $5,000 grant for lab supplies and equipment made possible through the generosity of the Boger Family Foundation. The winner of this year's award is Essex Technical High School. Located in Danvers, Essex Technical serves students from 17 communities in its region. Theirs is a true success story, and we at MassBioAD are very happy to have played a small role in that success. As I call forward their teachers and students who are going to tell you that important story, I want to recognize Kathleen Holman, who is the Director of Career and Technical Education at Essex. Thank you for joining us. And I also want to ask Dr. Michelle Mischke, our Director of Biotechnology Education at MassBio, to bring the award forward. As I call our speakers forward, it's my honor to introduce our Elisa Laporte and Deb O'Reilly, two incredible teachers at Essex Tech, as well as students Nadia Reed, Haley Martiniello, Maya Toribio, to accept this award on behalf of Essex Tech and to tell their story of success. Please join us up on stage.
My name is Elisa Laporte. I'm a biotechnology career tech teacher at Essex Technical High School. First, I'd like to truly thank MassBioAD for this wonderful opportunity. 4 short years ago, when Essex Tech first opened its doors, the Biotechnology Program was a few empty labs with a pile of boxes and supplies and equipment. The program started with one teacher with no teaching experience, who, much like many of you, worked in the biopharmaceutical industry as a research associate. To say I was overwhelmed that first day was an understatement. Then I met the students that I would be instructing over the next 4 years, and something occurred to me that was vastly different from my own experiences and perceptions of high school classrooms. These students wanted to learn. They were full of questions, and surprisingly, they didn't just want to know, they wanted to do. I often found myself not being able to ready the supplies and equipment fast enough because they were so eager to get started on their lab exercises and activities. This was a very different sort of education, but one with far more opportunity to engage students in creative ways and allow them to put into practice for themselves the concepts that they were tasked with learning. Essex Technical High School is a vocational and agricultural high school in which students spend half their time in their chosen career tech area, and the other half of their time taking the more traditional academic classes. The mission of Essex Tech is to create a culture of academic and technical excellence, encourage continuous intellectual growth, promote professionalism, determination, and citizenship for all students. I can think of no better way to do this than through an intensive, hands-on approach, one that truly immerses the student in the learning experience by allowing them to do for themselves the things they are learning about and apply the learning in a broader sense. Though Essex Tech is a school that centers on this idea, a school need not be vocational to provide hands-on learning to their students. Organizations like MassBioAD and Bioteach provide excellent workshops to train and educate teachers to use hands-on activities in their own classroom. MassBioAD offers high-quality curricula and truly makes it easy and affordable for educators to deliver these learning experiences to their students. MassBioAD offers exciting and engaging field trips, such as Biotech Futures and Career Exploration Days. We have been fortunate enough to be able to experience these events, and the benefit to our students is immense. They also help connect educators with needed equipment and supplies, as well as making connections for guest speakers to come into the classroom. They even offer desperately needed tech support when things don't run so smoothly. Both my colleague, Deb O'Reilly, and I have attended many of the BioTeach workshops, and we cannot say enough good things about the training and insight these workshops offer to educators like ourselves. We have implemented many of the lessons we have learned about in these workshops in several of our CTE classes, and have also recommended the workshops, activities, and online curricula to colleagues. Some of the activities that we have successfully implemented into our own program include having students explore their personal genetics by testing their PTC taster genotype and phenotype for the ability to taste PTC. We've also tested various food products for GMOs. We have performed polyacrylamide gel electrophoresis and Western blots to look for myosin in fish muscle. We have used indicators to test for different macromolecules. Other projects include transforming bacteria to express the GFP gene and isolating green fluorescent protein using hydrophobic interaction chromatography. MassBioAD has also made it possible for our program to pursue research projects like allowing our students to test DNA obtained from whole tick samples for the causative agent of Lyme's disease. We were able to do this by using nested PCR, and were able to further determine the subtypes of the organism in positive samples using restriction fragment length polymorphism analysis. Research projects like this allow students to use many of the different biotech skills and methods that they have learned in their CTE and apply them to answer research-based questions in an interest-driven project. This project has been a great source of engagement, success, and collaboration for our students who have shared this project with both our natural resources and environmental technology programs through poster sessions and meetings. Some of our students have gone on to use the skills and information gathered in some of these activities to present projects at both Skills USA and FFA, which is Future Farmers of America, contests. We have been working with our administration and fellow teachers to bring some of these activities and training into our academic classrooms as well. Our biotech seniors are working to bring some hands-on science into some of our district middle schools to get younger students excited about the potential of hands-on education in science and learn a little bit about biotechnology before they come to high school. Our program is still very new and growing. Our individual CTE classes have gone from 6 to 12 to 18 students, and we anticipate enrollment will go as high as 20 in the near future. There's still much we can learn and do with MassBioAD as a collaborator. We have many goals still to meet, but MassBioAD has helped and will continue to help make our program, as well as other classes at Essex Tech, more engaging, more successful, and more innovative. Thank you very much for choosing us for this award. We are very excited about what the future holds for Essex Tech and biotechnology. Thank you. I'd like to present Nadia Reed.
Hi, I am Nadia Reed, and I am a senior in the Biotechnology CTE at Essex Technical High School. When I was just a freshman, I wasn't sure what career path I was interested in, so choosing a CTE was difficult. After 8 weeks of exploratory, I decided on carpentry. However, I quickly knew it wasn't the correct choice for me. I knew I was more interested in health or science fields, and after speaking with my guidance counselor, I thought biotech would be a good option. Although initially I was unsure of what biotechnology actually was, I have learned so much about biotechnology than I would have thought possible in just 3 years. Being in biotech has ignited an interest in learning more about biology and chemistry, and I have become very interested in pharmaceutical sales as a direct result of my experiences in the Biotechnology Program. I have committed to Assumption College in the fall of 2018 as a business marketing major with a minor in chemistry to pursue my career in pharmaceutical sales. During my senior year, my class and I were able to use materials and supplies funded by MassBioAD, and through the support from MassBioAD, we were able to homogenize black-legged ticks and use PCR to detect a unique spacer region in the organism Borrelia burgdorferi, which is a microorganism that causes Lyme's disease. Using the tick samples collected by environmental technology and natural resources students, we were able to detect the presence of the organism responsible for Lyme's disease by nested PCR in 23 out of the 36 tick samples. Furthermore, MassBioAD supplied materials so that we were able to use restriction fragment length polymorphism analysis to subtype the detected microorganisms into 3 groups: RST1, RST2, and RST3. This project allowed us to put into practice many of the skills and methods we had learned in CTE, and some of the skills necessary to perform the tick analysis were nested PCR, gel electrophoresis, restriction digest, use of a bioanalyzer, and micropipetting. We were able to successfully analyze the results to determine which of the tick samples tested positive, negative, and had which RST types. I thoroughly enjoyed this project as it related directly to the real world and more specifically gave insight to how careful we should be when hiking in our area and examining ourselves for ticks. MassBioAD made this project possible by providing us with the funding to obtain the supplies needed to work on this project. In our CTE, we collaborate with other classes. For example, we ran a blood typing lab with health assisting using cheek cells. We have taught several of the AP biology classes to pipette and run agarose gels for DNA fingerprinting analysis of samples. And this year, we will be heading out to middle schools to introduce younger students to the field of biotechnology. None of these experiences would have been possible without the assistance of MassBioAD's help with the funding to purchase the Mini One gel systems and other equipment. I enjoyed being able to share our results about the ticks and ability to share biotechniques with other classes. I am looking forward to continue learning and applying my knowledge in biotech towards my pursuit of a sales degree in college in order to work as a pharmaceutical sales rep. And now I introduce Amaya.
Hello, my name is Amaya Toribio, and I am a junior in the Biotechnology CTE at Essex Technical High School. I selected biotechnology as my CTE because I am interested in becoming a hematologist. During freshman exploratory, I realized that biotechnology would give me the opportunity to practice skills and perform tasks that I may need to use in a hematology laboratory. I knew my experience in the biotechnology CTE would be more relevant to my career interest. At Essex Tech, our biotechnology CTE provides us with skills most high school and even college students may never have. We have transformed bacteria, prepared solutions of different concentrations, amplified DNA using PCR, inoculated bacterial cultures, and performed many types of separation techniques, such as column chromatography and gel electrophoresis. In a typical lab classroom setting, students sit down, read, take notes, and discuss concepts about biotechnology. In CTE, being able to participate in active labs have given me a unique insight to the vast biotechnology industry and how I may contribute to it. Practical experience acquired through our labs helps us picture ourselves as biotechnicians so that we can get a real sense of working in industry. We are able to plan and execute experiments and make observations and analyze data, giving us a more practical insight to real-world applications. With the support of MassBioAD, we have been able to complete many new labs and gain experience and simulate projects that really relate to the biotech industry. Even though I'm still planning on pursuing a career in hematology, I've gotten the chance to culture bacteria and isolate proteins, experiencing other career pathways thanks to some of the experiences made possible by MassBioAD. For instance, MassBioAD provided our instructors with the training to incorporate lab exercises in which we purified green fluorescent protein from E. coli transformed with P-glow using hydrophobic interaction chromatography. Projects like this prepared us for other projects like the Amgen Biotech Experience, where we processed through a series of lab exercises to engineer E. coli to produce red fluorescent protein using recombinant DNA technology. During these labs, I was able to gain hands-on experience in producing functional proteins from genetically modified bacteria. I learned about insulin and how it is made to support patients with diabetes. These experiences increase interest and build student confidence in science through understanding of biotechnology methods. In addition to introducing students to many career pathways, in biotech, MassBioAD has given us the opportunity to tour research institutes and colleges that have departments specializing of relating to biotech, such as Bridgewater State University and Whitehead Institute. At Bridgewater State, we are given a wonderful presentation from the Mini PCR co-founder, Sebastian Graves. We learned about how the Mini PCR was created and how it was used to solve problems during the Ebola outbreak in Africa. Graves also introduced us to a project called Genes in Space, which is a competition for high school students to come up with a potential lab experiment that uses the Mini PCR while in space. If we had not attended the conference at Bridgewater, I would have never learned about entrepreneurship and genes in space. The conference taught me that biotechnology has the potential to be used not only on this planet, but beyond. At Whitehead Institute, we received a presentation on electron and fluorescence microscopy. This experience gave us the ability to see new innovative technology that we do not have access to in the classroom. Also, we talked to Amelie Raz, a woman who researches regeneration of organisms. She taught us about planetarians and how they use signals from the muscle to guide regeneration. Lastly, at Whitehead, we met with Jing-King Wang. His presentation broadened our horizons on biochemistry. Wang showed us that science does not just happen in a lab, but all over the world. He talked about how he has been able to travel around the world to work with his team of dedicated scientists to research a variety of plant characteristics that could potentially help the human race. Without MassBioAD, my classmates and I would not have gotten the opportunity to gain this amazing knowledge of real-world biotechnology applications. Thank you. And now I introduced Haley.
Hello, everyone. My name is Haley Martinello, and I'm a senior in Essex Tech's Biotechnology Program. In the 4 years I've been in this program, I've learned a variety of techniques and skills that have helped me define the career pathway that I intend to follow. Biotech has introduced me to skills such as culturing hamster ovary cells and PCR analysis to identify Borrelia burgdorferi. Next year, I plan to attend the University of New Haven in the fall to focus on both biology and forensic science, and I can sincerely say that the education and experiences I've had in my class have solidified my career path with confidence. My biotechnology program has taught me many skills, but those that have made a lasting impression and stood out to me were labs focused on genetically modified organisms and labs focused on personal genetics. We completed a lab that allowed us to test processed food sample DNA for sequences indicating that they have been genetically modified. We used PCR and gel electrophoresis to accomplish this. This lab was introduced to both of our biotech teachers at a MassBioAD workshop, and the teachers were able to perform this lab and optimize their skills in order to teach us students how to perform the same lab. This lab introduced me to GMOs and their positive impact on the human race, exposing to me and other students one of the many footprints science leaves on the world. Another lab that has left an impact on me was a lab titled Personal Genetics, A Matter of Taste. This series of exercises was interesting to me because it allowed us to use our DNA from our own cells to make a discovery about our own genetics. In this case, we were using PCR to amplify 2 SNPs within the TAS2R38 gene, which is involved in the ability to taste PTC, which is phenylthiocarbamide. It has a bitter taste and can be found in foods like broccoli and Brussels sprouts, which nobody likes. And in common day laboratory work, fingerprinting is used to test for certain traits or sequences, and in this case, it's the gene influencing the ability to taste PTC. 75% of people have the ability to taste PTC, and the ability to taste PTC is encoded by the gene TAS2R38. So this lab included scraping some cheek cells from the inside of our mouths with a toothpick, centrifuging to get the DNA, isolating the DNA, and then performing some PCR amplification of the 2 SNPs that we found in the gene. And then we performed a restriction digest of the PCR products, and then finally, we ran the digested products on a gel. This lab was of interest to me personally because I've developed an interest in forensic science, particularly in subjects like DNA profiling, and the methods we used in this lab are applicable to that technology. Both of these labs that I have described have had a positive impact on our education and have helped us make important decisions about pathways to the careers that most interest us. It is because of the training that MassBioAD has offered our teachers that we have had the opportunity to experience these labs and a degree of learning that cannot be obtained by sitting in a classroom, reading texts, or being lectured to. Thank you guys so much for having us. It was great talking to you all.
Great job, fantastic job. Last year, MassBioAD programming through BioTeach reached 11,000 students, 98 different public high schools in Massachusetts. 11,000 students. Let's get to 20,000 students in 2 years. We can do that with your help. We are so thankful for your support, and let's hear one more time for these wonderful teachers and students.